we will learn about respiration. Now, what is respiration? So, it is defined as the process in which organic molecules are broken down to a series of chemical stages to release chemical potential energy. And this chemical potential energy is used to synthesize the energy-rich molecule adenosine triphosphate. Now, what kind of organic molecules can be used to perform the process of respiration? It can be a carbohydrate, which is usually a glucose molecule being used as a respiratory substrate. Some cells, such as red blood cells or lymphocytes, they use respiratory substrates as fatty acids or else glycerol, and sometimes amino acids. So these two are the respiratory substrates other than carbohydrates. The main fuel for respiration is carbohydrate, which is usually glucose. Other sources may include fatty acids and glycerol, together called fats, as well as amino acids. Coming to the glucose metabolism, now how do we get an ATP molecule from these respiratory substrates? So talking about glucose, the glucose passes through a series of stages, which is the glycolytic, the glucose metabolic pathway, through which it gets converted into this energy-rich molecule. Now, the stages of glucose breakdown are basically 4, and these stages occur in the presence of oxygen. So, respiration is usually of two types, depending on the availability of the oxygen. If the respiration occurring is in the presence of oxygen, then it is termed as aerobic respiration. And if it is occurring in the absence of oxygen, then it is called anaerobic respiration. So the stages I discussed here, the 4 stages, these are all parts of aerobic respiration. The first one is glycolysis, then we have the link reaction, Krebs cycle, and the last one, oxidative phosphorylation. So when the glucose, the product formed in these processes, they enter phosphorylation, the oxidative phosphorylation, the aerobic respiration ends here. It is important to know the location of these processes where they are occurring in a cell, in a particular cell. This is an illustration of a cell. Let's suppose this is the cytoplasm of the cell. And this whole purple thing is the mitochondria. So, from the diagram, we can clearly see that glycolysis is a process, is a part of aerobic respiration that occurs in the cytoplasm of the cell. Whereas the product formed by the glycolysis, it enters the mitochondria and it takes part in the link reaction of the cell. The link reaction forms a product, which then goes through the Krebs cycle, and finally the Krebs cycle proceeds to the oxidative phosphorylation, after which it is stated as the end stage of aerobic respiration. Okay. Then coming to the first stage of the aerobic respiration, which is glycolysis. Now, as the name suggests, glyco, which is used for a glucose molecule, and lysis means breakdown of any substance. So here, it is getting glucose breaking down into a molecule called pyruvate. Now, the definition of the glycolysis is that it is the splitting of glucose into 2 molecules of pyruvate, each containing 3 carbon atoms. As we know that glucose is a 6-carbon compound. Now, when a 6-carbon compound breaks into 2, each compound formed will have a total of 3 carbon atoms. So, these 3 carbon atoms we have as pyruvate molecule. We have 2 molecules of pyruvate. Pyruvate is a 3-carbon containing compound. So, the breakdown of glucose forms 2 molecules of pyruvate. Each molecule of pyruvate is of 3 carbon atoms. To proceed the reaction of the process of the glycolysis, energy-rich molecule ATP is required to start the process of glycolysis. That is, in the initial stage of glycolysis, we require some amount of energy, whereas energy is produced in the last stages of the process. So, we have a long process of glycolysis. In the initial of the stages, we will require some amount of energy in order to proceed the reaction, whereas we will begin to produce energy in the last stages of the process. Summarizing here before the start of the whole process, that we will get at the end of the glycolysis cycle, we will get a net of 2 ATP molecules, adenosine triphosphate molecules, per glucose molecule breakdown. So one glucose molecule breakdown will produce a net of 2 energy-rich molecules, ATPs. Here, we have the cycle of the glycolysis. It is showing. Now, let's talk about it step by step. It's showing that we have the first molecule, our respiratory substrate, as glucose. As we know that it is a 6-carbon compound, and we have 1 molecule of glucose, which is entering the cycle of glycolysis. The first step is that glucose gets converted into its active molecule, active form, which is glucose 6-phosphate. Now, how this compound is being formed? That is, a molecule of ATP, it gets dissociated into its constituents, ADP, adenosine dinucleotide phosphate, and an inorganic molecule. This inorganic phosphate gets attached to the carbon number 6 of glucose molecule. So we simply know that glucose is a hexagonal structure with 6 carbon atoms. So this inorganic phosphates get attached to the carbon number 6 of the glucose molecules, forming its active compound glucose 6-phosphate. The addition of phosphate to a substance is known as substrate level phosphorylation. Phosphorylation is simply the addition of phosphate to a substrate. Okay. So in the first step, we have the phosphorylation of glucose taking place. Then what's happening is glucose 6-phosphate undergoes molecular rearrangement and it forms its isomer fructose 6-phosphate. How does it happen? Glucose is a hexagonal shaped structure with 6 carbon atoms, whereas fructose is a pentagon. The fructose also contains 6 carbon atoms, but we have the difference in the structure, the shape of the structure. Glucose is hexagon and then fructose is pentagon. So what happens is that molecular rearrangement occurs, rearrangement of this carbon atoms occur, and what happens is resulting in glucose 6-phosphate getting converted into fructose 6-phosphate. So in the second step, we have the rearrangement of the atoms forming an isomer of glucose 6-phosphate, that is fructose 6-phosphate. Then in the third step, what is happening is again phosphorylation is taking place. Now, how do we get to know that phosphorylation is taking place? We can say here clearly that an ATP molecule is getting dissociated into its constituents, ADP, and an inorganic phosphate is formed. Now, what happens is that inorganic phosphate, it gets attached to the carbon number 1 of fructose 6-phosphate, forming a compound named as fructose 1,6-bisphosphate. What's happening is this stage is named as second phosphorylation. As here we can see that an inorganic phosphate is getting added to the substrate. So we can say second phosphorylation of fructose 6-phosphate forming fructose 1,6-bisphosphate. Then in the next step, fructose 1,6-bisphosphate. It gets, I'm writing it here, fructose 1,6-bisphosphate. This is a 6 carbon compound, right? First we had glucose, which is a 6 carbon compound. Then we have glucose 6-phosphate. It is also 6 carbon containing compound. Then fructose 6-phosphate. It is also an 6 carbon. And then fructose 1,6-bisphosphate. It is also 6 carbon containing compound. Then what happens here is in this step, fructose 1,6-bisphosphate, which is 6 carbon containing compound, it gets broken down into 2 compounds, each containing 3 carbon atoms. One compound is called dihydroxy acetone phosphate. And the other one is called 3-phosphoglyceraldehyde. Then what happens is this compound, abbreviated as DHAP, it immediately gets converted into another one. So at first we have a 3 carbon containing compound called 3-phosphoglyceraldehyde and DHAP. 3-phosphoglyceraldehyde is abbreviated as PGAL. Okay, so we had 2 different compounds, but then what happens that DHAP immediately gets converted into 3-PGA compound, 3-phosphoglyceraldehyde. So here in this step, we have 2 molecules of 3-phosphoglyceraldehyde. This stage in which fructose 1,6-bisphosphate is getting converted, it is getting broken down into 2 molecules, 3 carbon containing molecules. This stage is known as cleavage. Then proceeding to the next step, what is happening here is 3-phosphoglyceraldehyde. First, we have a removal of hydrogen taking place. So what's happening here is that 3-phosphoglyceraldehyde, it is removing hydrogen. And now what happens to this hydrogen is we have some molecules which are termed as hydrogen carrier molecules, such as NAD. It is a hydrogen carrier molecule. It gets reduced with this hydrogen and forms a compound NADH positive. Or we can simply say reduced NAD. So it gets reduced with this hydrogen. And then, so this step is known as a dehydrogenation. Therefore, first, dehydrogenation of 3-phosphoglyceraldehyde takes place. Secondly, an inorganic phosphate from the cytoplasm of the cell, it gets added, which is known as phosphorylation. So 2 things happens to this 3-phosphoglyceraldehyde. The first one is dehydrogenation taking place, and the second one is phosphorylation. So After these two things happening to the 3-phosphoglyceraldehyde, it forms a compound known as 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate, this inorganic phosphate gets attached to the carbon number 1, thus forming 1,3-bisphosphoglycerate. So here, two things are happening. First one is dehydrogenation. D means removal, and hydrogenation, removal of hydrogen. And the second one is phosphorylation. Okay. Then this 1,3-bisphosphoglycerate. So here we have formation of ATPs. 2 ADP molecules from the cytoplasm of the cell, plus this 1,3-bisphosphoglycerate. We have these 2 molecules of this 1,3-bisphosphoglycerate. So each molecule removes an inorganic phosphate. Each molecule of this 1,3-bisphosphoglycerate removes an inorganic phosphate. Therefore, we have 2 inorganic phosphates. These 2 inorganic phosphates, they get attached with this 2 ADPs, which we had from the cytoplasm of the cell, forming 2 ATP molecules, 2 adenosine triphosphate molecules. So here we have our first 2 molecules of energy being formed, 2 ATP molecules. After removal of this phosphate from 1,3-bisphosphoglycerate, they get removed from the carbon number 1, from which at which they get attached in the former step. So now these 2 inorganic phosphates get removed from the carbon number 1 from each molecule of the 1,3-bisphosphoglycerate, leaving behind 3-phosphoglyceric acid. So in this step, we can say that the removal of inorganic phosphate is happening, and most importantly, we know that formation of ATP is taking place here. Then we have the compound as 3-phosphoglyceric acid. This 3-phosphoglyceric acid, the 3 means that phosphate is attached to the carbon number 3. What happens is, again, rearrangement of atoms takes place, and this phosphate which was attached in carbon number 3, it gets attached to the carbon number 2, forming the same compound, but here the number is changed. Carbon number 2, 2-phosphoglyceric acid. So rearrangement of atoms takes place. Then we have the compound 2-phosphoglyceric acid. What happens is, here is dehydration of 2-phosphoglyceric acid happens. Dehydration. D means removal, hydration means water. What we studied earlier was dehydrogenation. Dehydrogenation means removal of hydrogen. Here we are talking about dehydration. It is removal of water. So dehydration of 2-phosphoglyceric acid takes place, forming 2-phosphoenol pyruvic acid. This is the second last step where dehydration is happening, and then 2-phosphoenol pyruvic acid removes. We know that we have 2 molecules of 2-phosphoenol pyruvic acid, each molecule removing an inorganic phosphate. These 2 inorganic phosphate gets attached with this 2 ADP, which came from the cytoplasm of the cell, forming another 2 molecules of adenosine triphosphate molecule, ATP molecule. And then after the removal of the inorganic phosphates, we have 2-phosphoenol pyruvic acid getting converted into pyruvic acid. So this pyruvic acid, it is the substrate, final substrate, or we can say final product of the process glycolysis. Now, what was important to talk about was the energy yield. How many molecules of ATP we use, and then how many molecules of ATPs we get. So let's discuss about energy yield before ending our lecture. Let's count the ATP molecules utilized and then formed. The first molecule of ATP utilized was here. Then the second molecule used was here. So we used 2 ATP molecules in the start of the process. Let's write it down below. We used 2 ATP molecules. Now let's see how many ATP molecules we formed. Okay, so here 2 molecules of ATP were formed, and then 2 more ATP molecules were formed in the last step of the process. So we had 2 ATP molecules, and then 2 more ATP molecules in the last step being formed. Calculating it as net gain was plus 2 ATP molecules formed. So at the end of the process, we have 2 molecules of ATP, new ATP molecules formed. Thank you.